Welcome to the Johnson Lab Mouse Behavior Room. Using our equipment to both encourage and measure food consumption in our animals, we can learn a lot about their feeding behaviors. To see what factors might affect an animal's motivation to consume food, we've developed a behavior testing paradigm to study cue-potentiated feeding, which we call CPF for short. When training the animal in CPF, we play a noise, and at the same time, we make a sweet liquid available to them. By repeating this food-cue pairing dozens of times over several days, the animal begins to associate the delivery of food with the noise cue, despite the noise and the food being separate phenomena. Due to this association between food and noise, we call this associative learning. By the end of the training, the noise alone will signal to the animal that food is in the area, just like the Taco Bell reminds us of burritos and weak hot sauce. Important to note is that noises other than the one we paired with food delivery will not cause this reaction in the animal. Even if the noise is the only thing going on, the animal won't pay too much attention. Finally, since we are also interested in how activating certain brain areas might alter this feeding behavior, sometimes we use a technique called optogenetics to activate these brain areas. To prepare our mice for optogenetics, we first inject the brain areas we're interested in with a virus, which creates light-sensitive proteins which attach to that brain area's neurons. We then surgically implant a glass rod, an optrode, into their brain, which will ultimately allow laser light to travel into the brain, stimulating the light-sensitive proteins and activating the brain area. Now that we have this conceptual background, let's see how our equipment helps us carry out the CPF paradigm. These Skinner boxes are where animals stay while we're training or testing them. The noises we play come from the speaker on the left side of the box. The sugary liquid we use is a simple solution of sucrose and water, but we may use other sweet solutions in our lab as well. The sweet solution is squeezed out of the syringes by these pumps, which carry the liquid down thin tubes and into the Skinner boxes. The liquid arrives inside a magazine on the right side of the box and fills a small food cup. After a time, we remove the liquid using a vacuum, so the mouse only has access to it while our sound cue is playing. While the mouse is consuming the liquid, their individual licks are recorded, which gives us lots of information about their consumption habits. Finally, to stimulate our optogenetic mice, we send a signal from a signal generator to a laser. The laser beam travels down a fiber optic cable, is split in two, and is connected by these even thinner cables to the optrodes we implanted into the mouse's brain. Do the mouse's consumption habits change when the laser is active? This could suggest that the stimulated brain area plays a role in feeding behavior.